Thank you very much. The great honor to be invited to give a talk. So first of all, let me thank my co-authors and my fellow students at Purdue University, and also my former students, Jingyun Tang, right now in Berkeley National Laboratory, and Yueyan Jiang and Xiaoliang Lu, both of them are at Marine Biological Laboratory at Woods Hole, Massachusetts. They made a contribution to this study. There have been lots of studies to simulate the different kinds of observational data into modeling systems to improve the prediction, right, of the ecosystem modeling biochemical. So in this particular study, I would like to ask kind of three questions. One is how the uncertainty of the ecosystem biochemistry models can be constrained, right? And particularly I'm going to focus on how the parameters, right, so one of the uncertainty source can be constrained. And the second one was the constrained carbon fluxes in North America. I'll say for modest error because we use some modest data to constrain the spatial variability, right? And third one, what we can learn from these model data, right, simulation or fusion exercises, right, for better having ecosystem models in the future. We have been using this modeling tool for years, and then I'm pretty sure in this audience you probably have knowledge about this model. And then therefore, I don't want to spend too much time to elaborate, right, this detailed stuff. But just point out that this is tool, right, we're going to use to explore those questions. I just said that uncertainty sources from ecosystem model perspective, it will be three primarily. One is input, right, data, for sure, lots of uncertainty surrounding. And the second one is model formulation and structure algorithms, right, or representation of processes and mechanisms in those models, right. And then another one is model parameters, right, what kind of values you use. And then if you do model simulation, you can see that it's very uncertain, right. All those can contribute to your model simulation, whether it's projection or estimation, right? I'll start with the conventional parameterization approach to improve, right, so parameters, quantification, estimation, in order to have a model application for either longer, right, temporal scale or a larger spatial scale questions. One of the key philosophy we use for conventional parameterization is, it's no secret, right, to use annual pools in the fluxes of ecosystem, carbon, nitrogen, for example, particularly for TM model, right, in the past, right, because we lack of higher frequency data, so we normally, we have annual pools in fluxes. Through biomass harvesting and so on and so forth, right? So in old days, this is a typical approach we use to match, right, to optimize the parameters to match the model simulations against the observations, and then extrapolate those parameters to regional or longer temporal scale questions. And then you can imagine that you only use annual fluxes or pools, and then you probably you don't have information about seasonality or interannual variability, right? So, and consequently, in the last number of years, there's lots of research ongoing is to be able to use higher frequency data, right? Like eddy flux data, right? You have right monthly or daily and even half-hourly, and then the idea is to use, right, such a higher frequency data to be able to further constrain, right, your parameters, So therefore, the Bayesian inference approach has been widely used, right? And the key idea is to use existing information, right, to constrain, to give you the most likeliable, right, the values for your parameters, right, in this case. And then turn out is you just need to calculate, for example, likelihood function in order to get the posterior, right, probability. And then here you can see VTIs to be able to consider the observation against model simulations, right? That's how the data being used in this procedure. Yes, for details, technical details, yeah, we have a former student, Jing Tang, published a number of papers in JGR in 2008, 2009. You can look at those technical details. Okay, this work is extended that, right, use that existing Bayesian approach to parameterize, right, those key or representative ecosystem types in North America, including different kind of forest ecosystems, grassland and tundra. And then an idea is to map these sets of parameters, right, to region. This example shows that how that Bayesian inference approach to give you the some confidence level, right, parameters values in some mode, right, so statistical mode values. And then we showed here is conventional, right, traditional calibration. And then using Bayesian approach, you have right, multiple sets of parameters, right, different levels, confidence parameters. This one shows the one example is how those parameters, right, being constrained to one site. You can see that we have several lines, TAM ensemble, and then you have a TAM, right, base conventional approach, and right, observation approach. You have mode, right, type of you can see that observation approach, the models, yeah, being further constrained compared against the observations pretty nicely. Right? And then you apply those sets of parameters, right? You got the ensemble, right? In this case, say 500,000 simulations because you have so many combinations of sets of parameters. Then we'll be able to quantify the, say in this case, net ecosystem production and distribution, right? And then also give you mode, most likely values. Compared to base values, you can see a conventional, right, approach give one value here, and the Bayesian approach give another value. In some cases, right, consistent, but in some other cases, even change the sign, right, of the sink in the source. So we say constrained, but not really constrained, rather, right, even larger, right? That's the problem. But anyhow, that the Bayesian inference approach, right, you need a large set of ensemble simulations, and it's not really efficient, right? And then in recent years, we further developed another approach, is the so-called adjoint approach, right? Again, I don't have time to elaborate that how the technically being done, but the key idea is again to efficiently calculate the gradient, right, in terms of the sensitivity, Fluxes sensitivity to parameters, and then use that information to further, right, constrain the parameters values by considering the Fluxes data. And then this using first 2 or 3 years, right, the adjoint approach, and then you got constrained parameters. You can see remarkably the model performed. So this is pretty amazing, right? So this particular case is one of the first site, and then we done this type of work for, again, for those representative sites. And another, therefore, we can be apply this adjoint approach because efficient. In this case, using models, right, GPP data, right, to using adjoint approach, right, to improve the parameters to be able to. For each pixel we have in this case, right, for example, Cmax is one of the parameters, and the rest of the other parameters associated with those ecosystem processes. So in the past, we always have one right, Cmax for each type of ecosystem. But in this case, you can see that Cmax actually is changing, right, from pixel to pixel, even the same type of ecosystems, right? So this is kind of it's new, different from our traditional way of doing things. If we simulate the models data first couple of years, and then in this case, in model parameters, right, constraint, and then use that, extrapolate those parameters, right, especially explicit parameters, you can see this 2006 models GPP versus simulated, right? You can see the magnitudes and spatial variability is very nicely matched. Right? And then this is, if we do aggregation for this region, right, North America, so in this case, we have three approaches. One is conventional, right? You only parameterize using annual flux and pools, right? Parameterize the model and do simple extrapolation, right? Versus using Bayesian, right? Ensemble, and then versus adjoint. You can see, yeah, they are very, very different, but this table probably do serve a better comparison, right? You can see that, yeah, GPP and NPP show the differences, and more importantly, you can see that native ecosystem production, TEM ensemble mode give you source of sink, right, and minus plus, and TEM base, which is conventional parameterization, give also sink, but adjoint TEM, right? Considering the models GPP, right, it's top for the constraint, the spatial variability in terms of parameters, and then rather give you a source, right, minus plus. So again, so this is not really constrained, right? Rather, it's uncertainty still even larger. Then we do nothing, right? And in conclusion, that in comparison with the a Bayesian inference approach, this is the new approach, adjoint approach, is more efficient, right? And then therefore we can be able to assimilate the spatial temporal satellite data, particularly as available, not just GPP, for example, other products, right? Vegetation, different indices, potentially we can be used to further constrain our model parameters. Then constraint, yeah, we're still using constrained carbon fluxes in North America. It's varying, right, range from a sink, right, 0.2 pentagram plus, yeah. So you can see that that's something even larger than mean, right, the value, and to a source about 20 teragram plus 0.13 petagram per year for this period. So through these studies, we can see that, yeah, so parameters you can do, you have a better control, right? So at start level, you can use those high frequency data. You can, those parameters could be well constrained, right? So, and even for spatial scales, right? For spatial, especially explicit parameters, if you're using sufficient information from satellite, right? And then you also can do a good job, right? And then for another uncertainty as input data, right, for ecosystem models, we don't have touch in here, but I think from these, right, different approaches, and then one of the biggest questions is that how we're going to, right, improve, right, the model structure or algorithms or representation of those processes and mechanisms in the ecosystem. I think that's still a big question, right? It's really big, right? Then, yeah, so in terms of acknowledgement, I would like to certainly thank the eddy flux observation community to be able to provide those very high frequency data, and satellite products development community also so critical, and also NSF CDI project provided funding to this work. have questions, and thank you very much.
Time for one quick question.
All right. Ichi, Professor. Uh-huh. What kind of parameters? All right, good question. Very, very good question. Yeah. So program design is flexible, right? Right now we have selected 4 or 5. I remember 4 or 5 key parameters. For example, one is Cmax, is always a gross parameter for that. Yeah, photosynthesis associated that, and several others in decomposition. Those key parameters associated with either carbon. Carbon assimilation versus decomposition. So those are, so indeed, so depends on how many parameters. If too many, then you got a problem. So this very good question, technical questions. Thank you.
Thank you. Our next speaker will be Ankur Desai discussing tall tower observations.
It's on the Mac side.
Yeah. How do I get rid of this?
What's that? You want me to do it? Good. How's everyone doing? It's Friday afternoon, 4 o'clock. Thanks for sticking around. It's been like me. If you're like me, I'm pretty tired. But it's been a really good week. 22,000 attendees. Lots and lots and lots of carbon cycle talks. So that's pretty exciting too. So I'm Ankur Desai, and thank you to Dr. Yi and Dr. Rashido for organizing this session. Looks like a number of good talks, and I'm going to talk a little bit about long-term flux tower time series. There's been a couple talks over the week about that, and there's going to be a couple in this session as well. And I think it's a really exciting time right now in the FluxNet community for being able to really start to look at these longer-term responses of ecosystems to climate anomalies. And so I'm not going to show any modeling today, just data, but I'm just going to kind of dive into it a little bit. And this is work that I just recently submitted to a special issue of photosynthesis research on photosynthesis in the environment. All right, so what's the motivation? And it's Friday, so I'm going to drink and talk. How do terrestrial plants respond to extremes, right? That's kind of a big open question right now. It's certainly an emerging major research area. It's something we've talked about for a long time, but I think a lot of what we're seeing, and then I gave a talk earlier this afternoon on frontiers in ecosystem science, where I made an argument that we're spending too much time trying to get our models to respond to the mean response, and not really focusing on these edge effects and the responses of extremes. And so this is just an image of the recent drought, of course, and this is here's an estimate. We had significant drought over most of the U.S., and there's this recent paper in PNAS showing the sensitivity of essentially the safety margin for when you have hydraulic conductivity loss as a function of mean annual precipitation. It was a really interesting, it's kind of this global convergence. And so there's some interesting teasers that suggest that there are consistent large-scale plant responses to extremes. And of course, another question we have is that a lot of our science, of course, originally in ecology comes from leaf scale work, plot scale, chamber measurements, and so forth. And a lot of what we really need to understand when it comes to how what's going on at the ecosystem is what's going on at the canopy scale, for at least for terrestrial forests. And in that case, there's a lot of features of the terrestrial system that don't quite correspond to what's going on at the leaf scale. That you can't, the linear scaling is not linear because you have things like self-shading, things like phenotypical variability in phenology, variability in carbohydrate storage, micrometeorological variation. All these things interact to cause variability at this larger scale to be quite different, possibly, than at the small scale. And this is interesting. So this is a so where are we looking, right? So this is a word cloud of every single talk title in the biogeosciences section this week. And clearly, there's a lot of interest in carbon. I was actually surprised how big carbon exceeded climate. And so At one level, I think we're maybe a little hyper-focused on carbon, but another, it really kind of points out an interesting area that there's a lot of interest right now in really solving this climate-carbon conundrum, both at the global scale and down to the stand scale. And we can see terrestrial forests, microbes, ecosystem dynamics, all of these things are really kind of coming to fore. So what do? Well, I do a lot of flux tower work, and I have a very tall tower. It's taller than yours. And it might help. It's 447 meters, and we measure fluxes of CO2, H2O, and methane, and a few others. And I'm just going to focus on the CO2 fluxes today. And the nice thing about a very tall tower is it can really start to estimate the canopy scale, stand scale fluxes. So this is just, here's a record. There's 1 year missing of fluxes. There's relatively weak trends in the data. So actually it's nice because it's mostly stationary, which allows us to really kind of look at the mean response over the long period. So what you see here is both the half-hourly as well as the kind of the 7-day average, or I think the 2-week average flux of CO2 and of water. So what I'm going to do with this data, so I want to extract a measure of productivity. I don't want to derive GPP using all these partitioning ways. I just want to look at productivity in some other way. So that's what I'm going to do. I'm going to try to see if I can identify some mode of variability, find some standardized anomalies, figure out whether these anomalies are how strongly autocorrelated are. And we'll see in a minute why that's an important thing to take into account when you're trying to understand how climate anomalies or extremes influence CO2 fluxes. And I'm going to test, do a bunch of lag correlation anomalies. It's far simpler than it sounds here. It's very, and try to just build some models just to see what's going on, just to get a sense of how might we go about improving models. So I'm going to do 2 tests. I'm going to assume that it's kind of essentially a hypothesis that positive lagged autocorrelation of productivity anomalies implies that there's a really strong internal, essentially negative feedback to response to extremes. So for example, this could be a feature of things like non-structural carbohydrate allocation, things that allow GPP to essentially persist in periods of extremes. And test 2 is that some timescales, moisture stress can overwhelm this internal feedback and lead to decreased productivity. These are just essential tests for scaling from leaf to ecosystem. All right, so first of all, there's a big issue with trying to get GPP from flux towers because we don't observe GPP or gross primary productivity. And many of us, including myself, have published just how sensitive it is to gaps and the type of model you use. So I'm going to do something a little bit more data-based. And so I'm just going to do this really simple test. I'm going to take the daily NEE, and I'm just going to take the maximum nighttime observed NEE. There's some papers from Van Gorsel and Ray Looning's group that showed that's a pretty good estimate of the mean respiration, with including accounting for advection. And I'm going to just subtract from the mean noon NEE. So it's just kind of, I call it photosynthetic drawdown, or PD. And I'm going to reject the noon NEE if it's more than 50% gap filled. I'm only going to use observed nighttime. So it's really going to give me as close as I can get to observations without worrying too much about gap filling. And so here's an image on the bottom of showing the PDs in blue. There's the night and the noon. And you can see it kind of looks like GPP. And so it seems like a useful thing to use. Maybe. And so I'm going to compare that to a whole bunch of other indices, like the Advanced Vegetation Index, evapotranspiration from the tower, water use efficiency, which is just, in this case, just a ratio of PD to ET, precip, soil moisture, temperature, minimum daily, maximum daily, diurnal temperature range, as well as modus terra on aqua land surface temperature. Okay, so first, I need to identify the modes of variability. And I could do this in some simple ways, and I'm not going to linger on this. This is an analysis that actually goes, it kind of is a tangent to much of this. So I'll just kind of briefly use this to describe why I'm using the averaging timescales that I am. So there's a whole variety of wavelet, Fourier type decomposition to understand spatial variability. And one of them that's really well designed for a lot of non-stationary gappy data and doesn't have that cone of influence problems with wavelet. And it's an empirically derived method, so you don't have to, say, specify a mother wavelet or any of that, is the Hilbert-Wong transform, which is based on empirical mode decomposition. Yeah, fancy acronyms and stuff. But basically, it's just a way to look at time frequency space of your signal. And what I show here on the right is that the time series of PD, my photosynthetic drawdown, and my ET. So on the y-axis is day length of wavelength, and on the x-axis is time over years from 0 to 15 years. And you can see that actually in this, what you see that's kind of like wavelets, this is much more of a kind of a stickier kind of approach. You're going to get really sharp relief in where the modes of variability are. And that's really useful. And on the far right is the integral overall across all time. And you can see that there's some characteristic variability, daily, weekly, monthly, and seasonal scales. The nice thing about this is that often with the wavelet analysis, you've seen papers from Baldacci and others in the past, some of those kind of greater than synoptic but less than seasonal scale modes of variability actually kind of pop out here as being more important than we usually think. All right, so pet peeve one, this is the first thing that we have to do. There's a lot of papers in our FluxNet community where we correlate one sine wave with another sine wave because they both seasonally vary with orbital forcing, like, I don't know, EVI and GPP, right? And it may not necessarily mean that one causes the other. And so I think it's important if we're looking at anomalies and extremes to actually take that data out. And the nice thing about having 15 years of data is I can just take out the ensemble mean. So what I do here, and this is just kind of my little figure showing the steps of going from the raw data on the top, which is my photosynthetic drawdown, estimating an averaging time scale, estimating first the anomalies by just subtracting off the long-term daily piece of D, averaging that over the selected time scale, and then just standardizing it by the standard deviation of the long-term variability. And that's what you get on the bottom, something that looks like a lot of noise, but it's actually a really useful, it's basically telling you how far above standard deviation is that day's photosynthetic drawdown relative to the entire 15-year record over some averaging time scale. Very useful, maybe. Okay, pet peeve two is that flux tower data, like most ecosystem process data, is highly, highly autocorrelated. And every time people do statistical analysis and they picture n as 17,520 or whatever number of half hours around a year, I want to hit them on the head because that's not the true statistical n for how flux tower correlations are likely to be. So I just used a simple approach from Brotherton, 1999. It actually comes from the climate science literature on like global climate model correlation techniques. Of just a simple thing. You're essentially just calculating the autocorrelation of your two variables across every possible lag. And as you can see here on the right, this is degrees of freedom for my 5,490 days of data for a piece of D. And you can see that as the averaging period goes up, it goes down to something like on the order of hundreds. Once you get down to looking at interannual anomalies, so this is smooth, you have to throw 365 days smoother across that whole time period, you're down to maybe single digits in significance, which really is what I'm going to show later, makes it really hard to test for interannual variability even with 15 years of data. All right, so what do you get? If you just look at direct correlations of every variable I talked about earlier to piece of D, and only the significant ones are shown, applying my little autocorrelation to estimate the n, the moisture and temperature anomalies do seem to positively correlate with piece of D, mostly at the subannual scales. And you don't see a lot of interannual correlations. And what's really interesting, ET and WE pop out. In some ways, that's a little bit of unfairness because they're both from the flux tower. And if you look at the actual MET variables, there's positive correlations with temperature out to about a 1 month time frame and soil moisture. Okay, that's nothing that we didn't know. It's where the lags get interesting, right? So now what I'm showing here is only significant correlations of piece of D with other variables. So A is piece of D with itself. So this is the autocorrelation matrix of the lags. And then in the other three plots, I show the correlations, and then the red squares are where those correlations exceed the autocorrelation of piece of D. So that's going to be my hypothesis test for where internal feedbacks are overwhelmed by other climatic controls. And you can see here something scary. One is most of you who do remote sensing science would think that enhanced vegetation index should correlate to some estimate of GPP positively. And here there's virtually no correlation at all at any time frame. So that's what's on the x-axis averaging period, and any lag, with the exception of these weak negative lags at like 2 months, which might be a phenology signal. Temperature doesn't do so well either. In fact, what's strong is the autocorrelation is really strong, especially the AR-1 lag, so the one lag prior to the averaging time scale. The sort of moisture lags. Now, this is probably the most interesting, at least so far that I've found. And so what I find is now if you look at ET, water sufficiency, precipitation, soil moisture, in the same exact format as the prior graph, if you look at lags on the y-axis, averaging period on the x, the earlier season, 2 to 3 month weekly to seasonal precipitation soil moisture anomalies strongly positively predict current photosynthetic drawdown at those same time scales. I have no idea what that means. But it's consistent with a lot of work that, for example, Dan Rasciuto showed in a paper several years back that interannual variability at this site was soil moisture related more than any other variable. And that's a puzzle given that what we're seeing here is something that's 70% forest, 30% wetland, northern hardwoods, relatively mesic, moist. The fieldwork is always buggy and wet. That doesn't make a lot of sense, but it does suggest that there's some interesting moisture-stress relationships going on that we're not picking up in models. It's trendy these days to do Granger causality, which comes from economics. So basically, it's just a way to kind of build models using all of the lags to see how much you can explain your current value. And so each of these variables on the bottom, what I'm showing you are the days, the lags that are significantly related to piece of D for different averaging periods. And once again, we just confirm that water use efficiency, ET, and all these moisture variables are having kind of between the previous day to the previous 2 months has a relatively strong or decent Granger-causing photosynthetic drawdown at this site. So that's a curious finding. I don't have a lot of mechanistic explanation for that. That's kind of the next step, but I thought it really points to how hydraulic limitation is actually a longer-term feedback on productivity than we typically think at the leaf scale or even a tree scale. So these are my thoughts/conclusions. So there's strong autocorrelation at the 1 lag for a piece of D, which supports a strong internal feedback at daily to seasonal scales. Soil moisture is important even in mesic forests, and we should be measuring it more and measuring it better, and precipitation, which we do terrible at most flux stats, are measuring well. Especially for early season moisture availability. So I didn't show this, but it seems like a lot of this is driven by the early season, which seems to impact then late season photosynthetic stress. And that maybe makes a little bit of mechanistic sense. 15 years of data, and this is a scary part, and I'm almost hesitant to put this conclusion up, may still not be long enough to credibly evaluate interannual to decadal scale modes of variability. And there's also some recent work by Harvard Forest that some of that is on that similar lines of thought, and I think we'll hear a little bit from Bill Munger later on what they find. Maybe they've solved it, which would be good. And remotely sensed vegetation indices may not be so useful for detecting GPP anomalies. They may be a really good instrument for picking out mean variability and spatial trends across large latitudes. And we saw this earlier when we did the big flux tower to MODIS comparisons, that we're very bad at picking up anomalies, but we're pretty good at picking up kind of large-scale variability. So things I want to do next, model evaluation and some multi-site evaluation, and talk to those of you who have flux tower time series that extend beyond a decade. So thanks. This is a paper that's submitted, and the funding from NSF. And I want to thank, of course, my lab members and my collaborators who helped make these measurements possible. So thanks.
Time for one quick question.
Trevor. You mean if I take out the flux footprint? Is that kind of, or correct? It is pretty heterogeneous. That's kind of why we're measuring there. And so we have this very small patchy wetland forest mix. And you're wondering if you go down to the individual stand scale, are those anomalies and drivers going to be the same? Yeah. Yeah. I see what you're saying. Possibly. It's relatively kind of fractal-like in the land surface, and so that doesn't seem to do a lot, but there is an issue of footprint bias, and that's certainly worth looking at. But I've never looked at it whether it's co-biased with climate. That's an interesting question. Yeah. Right.
Thanks, Ankur. Our next speaker will be Jing Chen.
Thank you. Thanks, Dan and Tree, for the invitation. I'm really very pleased to be here. And also thank you, the audience, for staying till this late in the afternoon, Friday afternoon. I'm very pleased to be talking, not only just our chairs, I thought it could be, and quite a few are here. For this session, I thought I'd try to emphasize on the climate part of our model. So try to assess the contribution of climate change to US forest carbon sink, in particular the Conterminous US Forests. I would like to acknowledge my co-authors, Famin Zhang. She was my student, now postdoc, and she did most of the computation. And our colleagues, Rich and Yudi, and they have been working very closely with us on this project. The motivation of this work has been that most of you have known the work published in Science last year, and the world forest sink, it is about 2.4 petagram of carbon per year, while U.S. forest consists of about 10% of that. It increased a little bit from last century to two centuries ago to most of the last century. And this sink could be related to various processes. And here I list some data sets that could be used for studying these processes. One is the forest age map. Is a compilation from various sources, in particular forest inventory data. This is published in BGC last year also. Then nitrogen deposition and climate change, and mostly it will be temperature and precipitation. Temperature, of course, increases most places, but southeast forests even decreased a little bit over the last 100 years, a little bit surprised to me. And while precipitation increases in most places, but there are also decrease in these Rocky Mountain area. Let me summarize what we are trying to do. We want to include these mechanisms in our model for the sea sink. First, the disturbance effects. That includes the direct carbon emission caused by fire, harvest, insect, and the regrowth of the disturbance. I'd like to point out that the regrowth part is included in the disturbance in our way of assessing the mechanism. And the non-disturbance effect, they are on both growth and respiration, includes climate, mostly temperature and the precipitation, and CO2 and nitrogen deposition. There are more than these, but these are the three factors that we consider, and a lot of studies involved with these various aspects. The questions we like to address is that what is the dominant driver for the sea sink, whether this is non-disturbance or disturbance factors. And among the non-disturbance factors, and that include climate, CO2, and nitrogen, how important are the climate factors, temperature and precipitation from the sink? And also, at the end, I would like to talk a little bit about is the changing climate important for the sea sink in the near future? First, I would like to start by showing a kind of brief list of previous studies in chrono sequence, with the early ones at the top, the latest one in the bottom. The numbers here in bold is national value. They vary quite a lot. But I would like to draw your attention to the fact that in the earlier studies, I could not seem to make it to work. Okay, the earlier studies more focused on non-disturbance effects, with recent studies more focused on the disturbance effect. Because the disturbance effect is more recognized as the main driver. And to do that investigation, we need a tool. And this tool should be able to consider both disturbance and non-disturbance factors. So this is called the integrated terrestrial ecosystem carbon model. This is to integrate disturbance, non-disturbance effects. And in the model, we have these factors considered, and in particular, we consider the regrowth part after disturbance, and of course the climate to assess the net primary productivity, and also we have soil line pools and consider some product pools, and most important is the vegetation carbon pool. All combined together, we can model net ecosystem productivity or net biome productivity, depending on the data variability. And the standard age is from the standard age map I showed. And in particular, we work quite a bit of time on the regrowth curve for 18 species groups from the FIA data. This is published last year in Global Biogeochemical Cycles. And we also like to address the uncertainties in both disturbance records and also regrowth records. This black curve here is what we could derive the total disturbance from the forest age map. You can go back from the age in the year of disturbance for each pixel. And these bottom lines, including fire, insect, and harvest, is from the real record. Also, this fire from Berger et al., it shows that in recent, about 50 years, they agree with what we derive from the inventory data from the forest age map, but does not agree with what happens 50 years ago. So what happened could be that there is a repeated disturbance from the same pixel. That is not picked up in the age map. So that is one big uncertainty. The second is this MPPH curve. This is the two examples of the 18. And they also have uncertainty. So we bracket about 20% variability. And so this one, for dealing with this difference here, we did this kind of model sensitivity test. And we can assume the remaining difference, or remaining which is not detected by the forest age map, we assign to either 20 years old, 60 years old, 100 and 150 years. See what these assumptions affect on the carbon cycle in recent years. Before I go into the results, I'd like to ensure you that we pay some attention to the validation. And these are the results, state-by-state validation for the soil biomass component. They are reasonable. We also have quite extensive validation against the tower flux data year by year. This is published in JGR last year, earlier this year. And now I can jump into the results. The total MBP is about 231 teragram. That's about 0.23 petagram. That's almost about the average of these two values, surprisingly close. And out of that, the disturbance effect occupies more than 80%, actually. And the disturbance effect, do I have pointer? Maybe I can use pointer. Okay. Yeah. The disturbance effect of the regrowth is the most part of that, and the carbon release during disturbance is smaller than that. But I like to draw your attention to this climate effect. Overall, is negative, while the other two effects are also relatively small compared with the disturbance effect. And if we accumulate it for the last 110 years, the non-disturbance effect takes about 18% here. It's about 18%, while climate effect is almost zero. And these three effects do not add up to the total disturbance because there are also their interaction. They interact each other, and generally interaction is positive. And just a little more detail about these two factors for precipitation and the temperature. Now this is accumulated, it's not year by year. It gradually accumulates with time. And this uncertainty line is due to the assumption of equilibrium. And the way we address the assumption is that we change the forest age at the beginning year for those pixels that are not determined well. This one is due to the age curve. They are more or less symmetrical along this mean value line that we only show the bottom one. The temperature effect is mostly negative, and this is also due to similarly due to equilibrium assumption and also due to the MPPH curve error. And it's negative. But I'd like to draw your attention to this one here. The absolute magnitude is much smaller. And then the total disturbance effect. Here is in terms of teragram, there is petagram. And so in region by region, we can see that they differ in some way. I do not have time to get into that. These region by region results, I'd like to draw your attention to the Rocky Mountain. The temperature effect is negative. Mostly it causes more drought. It's in already quite dry area. And for the future, I may not have time to get into that. I'd like to just to show you one final result that it is also small for the A2, the climate effect. Total is negative. For the B2 scenario, is also small, climate. But this is already, the summary is that the climate effect in the past and the future, at least in the way that we do the modeling, is both small. But I like to mention that we have not included the climate-induced mortality and successional changes into this one. So these, for the future, still is a preliminary result. Thank you all.
We could take one question.
Yeah. That's right, yeah, correct. Yeah, I already showed there. That's right, yeah. Yeah. And mostly it is recovery, regrowth here, mostly.
Thank you. Our next speaker will be Bill Munger, discussing the now 20-year record at Harvard Forest.
Well, good afternoon, and thank you all for sticking around, and thanks for the organizers. And I would start at the beginning and just make sure to acknowledge or the past students and postdocs. We have many people over the time period of measurement. I won't list them all. That would take all my time up. And thanks to the sponsors, particularly the DOE and or the NSF, which are the main contributors. So in a nutshell, what my goal in this next 15 minutes is, I want to identify and hopefully attribute some anomalies in the net ecosystem exchange and try to associate those with the climate anomalies or other events. There's a complication in trying to look at NEE because we're measuring NEE, and that combines multiple processes. So trying to attribute NEE to any one thing you have to be aware that say warmth might affect the uptake and the respiration part independently. And as I'll show you, the outcomes from a particular anomaly depend a lot on the timing of when that anomaly occurs and what the prior state was. And as Anchor has said, you will see lags. I won't show anything looking at lags, but it's important to consider that there's lags in the system, and you can consider legacies, which kind of goes together with what the prior state was. And so just quickly, how does net ecosystem exchange get influenced by the climate? One, you can have a big storm and it knocks down the whole, or the canopy. I'm not going to talk about that, except to note that the current vegetation that we see at our site is in part there because there was a hurricane in 1938, which largely knocked down much of the canopy. But fortunately, we haven't had a hurricane at our site since then. We've had near misses and some big storms, but nothing catastrophic. There's kind of an intermediate where the kind of climate effect that is the kind of mild direct forcing of the climate, which may largely be already included in the models, and so I'll mention we like to not. So I don't want to consider that in the extreme. There's certainly droughts and frosts and heat, changes in snowpack, and some of these may cause thresholds and tipping points. As I say, the kind of mild influences here, those may be part of what models already get. We won't learn anything by doing that. So what I'm really interested in is kind of what are the end-effect effects of feedbacks and lingering or the consequences. How does ecosystem function and change as a response that may have a more lasting impact? And some of the things to consider are, if you have a climate change, you'll change insect infestations. I'll mention, and we're probably seeing some of that. You may have a small storm will make gaps in things, and that not quite as severe as knocking down the whole forest. So sort of subcatastrophic effects. And how does climate affect the nutrient cycling, nutrient uptake, the capacity of plants to take up nutrients? So I think it's these kind of indirect and feedbacks that are really what we need to know more about. So we have long-term observations. Why is that useful? Well, one, the climate changes. The climate does change, but it's slow and has a large interannual variability. And in a forest, the vegetation and carbon pools, they have a considerable inertia, so they're not going to change very fast. If you just measure for a little while, you might not see anything happen. And the same combinations of wet and warm or cold or the timing of anomalies, it takes a while to see more than one of those. If you just get one event, you say, oh, well, such and such a thing happens when we get a warm spring, but you need several warm springs to be really sure that what you're seeing is and extreme events don't happen very often. We have one mild ice storm in 20 years, one hurricane in 75 years, and so on. So to see effects of those extremes, you really need to have a lot of data, and we won't see that on site. So yes, the climate at Harvard Forest is changing. This is the mean annual temperature at the meteorological station going back to when that particular station was established before 1970, and there is a trend about 0.3 degrees C per decade, and there's a very large interannual variability, which is bigger than the trend over that time period. But importantly, that mean annual trend doesn't really translate to, say, consistently warmer winters or colder. This is the blue line is the overall monthly mean, just repeated from year to year, and the little dots are the actual. So those really are the anomalies, sort of month by month, and they're smaller than this amplitude, and they don't sort of consistently show you that the winters have gotten warmer or the summers are hotter. So those annual variability in climate does not translate to a consistent change during any one time of year. and looking at the precipitation, again, that's the mean just repeated. There's a lot of anomalies in the precipitation, but not a consistent shift of consistently dry or consistently wet. There's not a sustained or systematic change in the precipitation regime. It kind of hovers around 100 millimeters per month, plus or minus. We see large excursions. The mean is around 100, and we see variability. So how are we going to define anomalies? Anchor has already mentioned one where you just subtract the full mean of the record. And in our analysis I'm doing here, I don't want to do just extracting the mean because I want to account for the direct climate response. So for the climate, I have just taken the overall mean. But for the NEE, we do something more. There's a number of ways you could do it. One is, as was done in Sean Urbanski's paper, is do a statistical fit to the observations that takes out the temperature response and the light response. So we're doing that so we can account. If there's a warmer summer, we don't want to know the anomaly just because it's warmer, or if there's a cloudy period, we want to take out the expected effect of having less light. There's other approaches, as we've seen, or the process model that Trevor Keenan has described, and there's also other statistical ways. So what we want to do is capture the predictable response and then look at how the residual depends. And the residual may still include some of the unmodeled parts that are in our model, moisture stress and things like that. But so one thing I can say is that if we look on the hourly step, any of these kind of models get a really good R squared. So just a simple fit to the data captures most of the variance on the hourly scale, largely because the model knows when the sun comes up and the sun goes down and when the winter is. But if we add those up to a year, and we don't, the black line is the observations on an annual, and these are two different ways to predict. It doesn't get it, and we have talked before about having a trend. Well, maybe it's not so much a trend as there may be we see this trend may have gone away, but it doesn't capture that either. So I'm not going to, and two kind of things to just point out. Two of the years that you'll see in the next few plots are in December 2008. We had an ice storm at our site. It knocked down some of the branches in the canopy. It didn't really cause as much of an impact as it did in other parts of the state. And we had 2010. We had a very, very wet March, a very warm April and May, and then it had a little frost, and then it was dry some summer. So you'll see 2009 season and 2010 a few more times. So the next, I'm not going to show you all of the different combinations of all the months and all the. So I can show you So on the bottom is the difference of the monthly temperature from the mean for that month, and this is the difference in the NEE from the projected. So if it's positive, there's less uptake, more respiration than you predict, and then negatives, more uptake. So one of the effects we do see is there's a fairly consistent, this turns out to be the largest this is the largest correlation of any of them, is that when you get warmer, when you have warm anomalies in the month of May, you get up to 60 grams of carbon per square meter per month more uptake. But that didn't translate to, so a really good May in 2010 didn't turn into a really good August, so the ecosystem forgot how well it was doing in the month of May by the time August came. It was still warm in August, but it was in fact kind of giving off more NEE than it was supposed to. One thing that accounts for that is if you have kind of consecutive dry months. So here's 2010. We see this is one of the driest or Julys. It had been very wet in the spring. It started off wet, started off warm. Everything got going, but then in July the rain kind of quit. So we see, in fact, less uptake, and by August there's still less uptake. and 2009 showing you here in July. Also in June, keep in mind that this is one of the wettest or the wettest months on the record. There's not too much of an anomaly here yet, but as you'll see. And the other point is, unless you have, there's not very much of a response to precipitation. And on the last one, I forgot to mention that in August, there's not very much of a response to temperature, except for some of the outliers, except for very dry. You have to have kind of consecutive dry periods to see very much of a precipitation effect. One dry month probably won't do anything. And I want to point out there's kind of that we have interactions between temperature and moisture. There's a weak September response of more uptake as it gets warmer, except for some periods that are, if it's warm but dry, as you see these are the same years, those are years where September was unusually dry, so it didn't matter that it was warm, or the vegetation still went into, or it still started to senesce. And then, as I said, 2009, this kind of is an outlier. You see there's a lot of uptake in 2009. So even though it was dry in September, it was very dry and cool, it still had anomalously high uptake. And I believe that's because the system had been charged with a lot of water during the summer, so the vegetation could keep going during that time of year. One of the main things we see that is the effect of climate we see is that leaf out is starting considerably early when we define, or the uptake season is starting earlier, and the uptake season is lasting longer, where we're defining this by the days that have net CO2 uptake. And when you look at the difference, the growing season has gotten considerably longer over the time period of record, and especially in the last 10 years. This just to show you that there's not very many large correlations on the monthly time step. So in conclusion, the time of year that the climate, or that a site like Harvard Forest seems most sensitive to changes in climate are in the ones that affect the end and the start of the growing season. Warming in the middle of summer has a very weak effect, and it's important, or at least in a site like or the Northeast, where the climate things change things change on a rapid pace, so we don't have consistent annual droughts. So the stresses in one part of the growing season may offset the next one. So we have a warm, wet spring, and got the NEE going, but then consecutive dry summer months undid that, and a wet June and a wet early part of the season in 2009. It was quite wet, and the people going out to look for the branches knocked by the ice storm got really soaked. But it meant that even though it was dry in September, everything still went or the forest was particularly happy. And so, as I said, that we did have an ice storm. It had a very modest effect. Spring frost and our footprint, those are our thought, and somebody might ask about those. We haven't seen one we haven't seen one at the right time of year at our site to have an effect. And 2012 was particularly an anomaly with very minimal snowpack and not very many cold temperatures, which we have seen has led the hemlock adelgids, which are in that area and will kill the hemlock trees. Those really did really good during the past year, so we may see that that kind of accelerates that infestation. And with that, I'll come to a close.
Thanks. In the interest of time, let's move on. Our next speaker will be Yichi Liu.
First of all, thanks to all for the opportunity, for your invitation. And also thank you for sticking around. Hopefully our talk is worth your time. And Well, in the past probably couple months, and we discovered a pattern. It's a globally very conservative, highly conservative pattern of annual GPP as determined by phenology and physiology. Today, I'd like to show you how we discovered that pattern and then get us your responses. But before we do that, I'll show you our motivation. And if you happen to be in the first session on Monday about the new mechanisms, new approaches for carbon cycle research, and you listened to the talk delivered by Su Li, probably you know that my lab in the past couple of years tried to develop a so-called traceability framework to facilitate the model structure analysis. The idea was that the simulated ecosystem carbon can be decomposed to a few components, and then those components can help to diagnose model structures for the model intercomparison and the benchmark analysis and data assimilation. We have very successfully studied the resonance time part and applied to the global model. Recently, we were thinking about what would be the simplest approaches to decompose the carbon influx. So kind of we look at the global map of simulated the GPP, and you can see very clear latitudinal gradients and gradually change from the tropic and become smaller towards the polar regions. If we look at the specific sites, carbon fluxes, so you can see these during the winter it's very low and then gradually increase and then decline. So if we say these, the maximum, seasonal maximum GPP represent the physiological processes, as like VC max or leaf air index, then this is so-called the phenology as represented by carbon uptake period. It's basically determined by climatic change. And so our question is how annual GPP is jointly determined by phenology and physiology along latitudinal gradients of the globe. And then we use the data source from the eddy Fluxes network, and also we use the modeling results to analyze the global pattern. So we use the method developed by Lian Hong Gu and to analyze the phenology and the physiology, and use this so-called calculated cap and this so-called stabilized phase of maximum GPP. So if we just plot the annual GPP against the two variables of cap and the GPP max, actually we don't see much of the relationship at all. And then we had an idea. If this is the maximum GPP and this is the cap, so if we time this, we get the rectangle area. And then this annual GPP is basically area below that curve. So we plot this annual GPP against that area, rectangle area, and then we get a ratio. So we find this ratio is so highly convergent to the value of 0.63. So that's from the 765 site year data, use the global database. So we don't know why. And then we try to explore, see if this relationship would be conservative for those long-term sites. So we picked those sites with eddy Fluxes measurement longer than 10 years, and to analyze basically every year. Then in this way we can see the interannual variabilities within one site and across sites. Actually, all those interannual variability and cross-site data all fall on the same line, 0.63. Then we analyzed these global model results from the Australia Community Atmospheric Land, Biosphere Land Model. And then we analyze this modeled annual GPP against the modeled that's cubic. And then this is very conservative again. And only the major outlines are here. This outline is basically those tropical forests near the equator, tropical regions. So basically, we basically lump all those data together and the model results together against the different biomes. And the tropical forest is in this region because it's understandable being outlier because it's not very clear those seasonality. But for all other biomes, as long as the seasonality is very clearly displayed and all fall in that line. So we are thinking about why the nature gave us such a highly conservative pattern. So that's the question we are still pondering on that question, and we are still thinking about, we are exploring different explanations. So here is the two of the ideas we have explored. Let's say a system, ecosystem, the phenology didn't change. But somehow physiology changed, become higher or lower. In order to maintain that conservative ratio, then the system has to have the GPP increase much faster for the higher maximum GPP, and drop much quicker in fall. So likewise, if this maximum GPP low, this is supposed to be slower. So then we're supposed to find some relationship between the maximum GPP and then the speed of this recovery or senescence. So that's the one way to examine the conservative relationship. So another conservative mechanism that could maintain that conservative relationship is through change the so-called the width, the basically shoulder. Let's say if the maximum GPP doesn't change, but the phenology changes, seasonality changes, then in order to maintain that conservative GPP, the ratio, then the shoulder have to be wider for the longer seasons. So we look at the data again, and find this relationship. It's not that tight as the 0.663 relationship, but the relationship there indicates that nature probably does have the mechanisms to be compensatory to maintain that ratios. So that's basically what we have found, and we have examined this relationship in a variety of ways. Seems like it's a very conservative pattern, and we have examined the different approach, different mechanisms why the nature maintains that such conservative relationship. If this relationship is really true, then we can use this relationship to do lots of things that probably before we are unable to do. I'd like to get your feedback. Thank you very much. Oh, I have lots of time.
Plenty of time.
Jane. Well, what the value? GPP maximum, we use the Lienhong-Goose method. Basically, this is the method we used. Let me see if I can get this right. We tried a different way to calculate the cap and the maximum GPP, and we found that Lienhong's method probably is the best one, and the most reliable. I mean, the reliable method. And basically, this not pick it doesn't pick one day. Just use these Lianhong developed the so-called stabilized phase of maximum GPP. And we also used a different way to calculate, actually, before we used the Lianhong's method. And we found actually the other method is not as stable as this one, but the general relationship still exists. 0.63 relationship. Qinglei. Lianhong, you are the expert. Help us to answer. Qinglei.
All right, quick. Could you elaborate how you said that could have lost usefulness with this relationship?
What?
So, for example, if you know phenology and if you know the maximum GPP, is this thing you can get any GPP?
Well, if you know the sites changing like GPP, maximum GPP, and also phenology, the growing season length, you just time 0.63. You get the yeah. And also this has a lot of implications because actually we are currently exploring one thing is it could be used to our so-called traceability framework. Another implication is actually this, we are exploring mechanisms. This 0.63, it's just almost exactly the number of 0.63662 of that sine curve. So if the sine curve is, if the biology follow that climatological space, bioclimatological space, then that means different vegetations just use the capabilities, fully utilize the bioclimatological resources. Then those models use those dynamic vegetation become less useful. I mean, especially for carbon cycle, specifically for carbon cycle research. I didn't say that the dynamic vegetation model not useful for any other purpose, just for this GPP per se. Dynamic vegetation model can be useful for lots of other purposes, but for this probably, it's if we have such conservative relationship, it could be.
Thank you, Ichi. Our next speaker will be Christopher Schwalm. He'll be discussing continental scale drought.
Hello, everyone. Thank you for sticking around on a Friday. What I wanted to talk about today was is a large-scale drought event which happened in Western North America between 2000 and 2004. And there are actually three things that I'd like to do here with this. First, I'd like to give you a little bit of intuition as to the drying down of the terrestrial biosphere that then happened from 2000 to 2004. The second thing is to effectively map what I'll generically call the water consequences onto, to map the water consequences to carbon. Okay, sorry. Okay, to map the water consequences to carbon consequences. And then the final thing is to really try to understand what the likelihood of this 5-year event was in the past and how that's going to change in the future. So let's start off with a very simple spatial representation of the so-called turn-of-the-century drought. So this is the drought monitor product. And so we have 2000 to 2004. The only thing that I'm showing here is the month of August. But what should be very, very apparent here is that the vast majority of the land surface here is in severe, extreme, or exceptional drought. So I would like to show you now a series of what I'll call indicator variables so that we can have some better intuition as to the actual temporal dynamics of this particular drought. And it turns out that in order to do that, we will simply contrast 3 years before the drought and 3 years after the drought with the actual 5-year drought event. So it's this notion of having a so-called neutral baseline. So at the top we then have the Palmer Drought Severity Index, then we follow with precipitation, and then we follow with soil moisture. One thing should be fairly apparent here is that these are all independent sources, right? Completely independent streams of data, but they actually show the exact same signal in terms of the turn-of-century drought. That is, they all see this 5-year event in the same way. And basically what I mean by that is there's a very clear negative excursion relative to the reference line. We can see the exact same thing in this other set of these so-called indicator variables. This set is a bit more eclectic. We can start with the top here and look at evaporative fraction, which of course speaks directly to the water cycle. It also speaks to the partitioning of available energy. We see a similar signal with regards to satellite greenness, a similar signal again in terms of evapotranspiration. And I also wanted to show you this, where for 4 of the 5 years we actually have a positive excursion, meaning that during those particular years it was actually warmer there. Now, if you think about all of these, again, what I've generically called water consequences, one good way to really integrate all those is to look at water availability. And that's exactly what I'd like to show you now. So water availability for this particular slide here is basically defined as surface runoff. And these are the five large basins within the western United States. And what you're looking at again is a time series here. And this is then the change in change in runoff expressed as a percentage relative to the 11-year mean. And the same type of precipitous decline relative to the turn-of-the-century drought that we saw in all these other indicator variables is also present here. Let's do, in effect, the exact same thing, but take a slightly longer-term view. That is, there really isn't any reason per se to be wed to the 11-year period. And how I'd like to do that is to then look at runoff percentiles, which then go from 1901 to the year 2010. So we have 110 years of data to actually work with now. And with a partial exception of the first year of the turn-of-the-century drought, what should be fairly apparent here is that the majority of the land surface is here or actually lower, which means we are either in the lower quartile of this particular runoff distribution or in the lower decile. And if you look closely, although these two colors are unfortunately rather close, you can can actually see there are fairly significant swaths where this is the lowest runoff year in the actual historical record. One final thing that I would like to do, in really keeping with this spatial depiction, is to just briefly show you this notion of spatial coherence. So drought has the reputation of being a very patchy phenomena. But what we see here in looking at these three indicator variables, again, precipitation, soil moisture, and palmer drought severity index, is that it's actually fairly uniformly distributed across the entire study area. The actual major exception is, of course, here. So there's this notion of spatial coherence. So again, this notion of starting with water consequences and then mapping them onto carbon. I'd like to stick with this explicitly spatial representation. And there are, of course, many ways to generate spatially continuous fields of these various carbon fluxes. What I'd like to show you initially is an upscaled FluxNet product. And what you should note is that as we saw in the previous slide, this is really a kind of uniformly red blob. And basically what I mean by that is that GPP was very much negatively impacted. Ecosystem respiration also, and then the reason we have a degradation in land sink strength is because the response of GPP was greater than the response of ecosystem, of total ecosystem respiration. Let's look at this from a slightly different perspective, namely using the MODIS products of GPP as well as NPP. Now, this is not quite a one-to-one comparison because we do not have MODIS data prior to the year 2000. But despite that, there is still this high degree of spatial coherence here. And the main point that I'd like to emphasize is this notion that plant productivity was very much depressed during the turn of the century drought. Let's change scales. And what I mean by that is instead of really looking at the half degree as well as the 1 degree products that we've been looking at up to this point in time, let's actually look at individual flux towers. And there are hundreds of these and a few thousand site years worth of data, but for purposes of the turn of the century drought, we actually have 15 towers where we have data that were spatially as well as temporally coincident with this natural experiment, namely the 2000 to 2004 drought. So what I'd like to do then is just show you how FluxNet saw this particular event. And how I'm going to do that is to take all 15 of these FluxNet sites and then first aggregate them by plant functional type. So these green bars are the evergreen needle-leaf forest. We have 8 of those, and then we go to grasslands where n equals 4, and woody savanna where then n equals 3. And what you should note here is really very much in parallel to what I showed you previously. Both GPP as well as ecosystem respiration were very much negatively impacted, but the overall response of GPP outstripped that of ecosystem respiration, and the order of magnitude here is about 60 grams of carbon. Note that if we look at the woody savanna site, we actually see a slight enhancement. This is actually driven by a single site. So 2 of those 3 sites are very much in line with this overall pattern, but we have one anomalous site that one anomalous site which in effect serves to flip the sign here. Let's briefly consider a couple of other carbon balance terms. Crop yield, which is really another way to talk about NPP in any type of agricultural system. And this is also a time series plot similar to what I've shown you many times here. And you'll note this exact same type of signature, where there is a clear negative excursion relative to the actual reference line here over this 11-year period. And it turns out that agricultural productivity in the western United States during the turn of the century drought was actually off by about 10%, and in the year 2002 it was actually depressed by about 15%. One other carbon balance term that I think we should really well, that definitely needs to be brought up in the context of any type of long-term drought is of course fire. So this is then data which then comes from the so-called MTBS So that's the monitoring trends in burn severity. Note here that we have kind of a, let's call it long-term mean from 1984 out to 2008. And of course, during the turn of the century drought, there was a much larger, or there was a larger area that indeed burned. And this, of course, maps directly onto fire emissions. Here we have our, again, long-term mean of about 10. And during the turn of the century drought, the actual average here, again, there's a fair amount of, let's call it intra-drought variability. We were actually up to about 25, which means that the enhancement effect here was in the order of magnitude of 15 teragrams of carbon on an annual basis. So let's just briefly step back now and kind of survey all of these various data products that I've used, particularly in the carbon side of things, to really understand the net carbon consequences. And what should be very clear here is that what happened during the, what happened during the turn of the century drought was that the land surface here really was significantly degraded in its ability to scrub the atmosphere of CO2. And it turns out that if you contrast all of the various anomalies that I have presented, if you contrast those with a yet completely different estimate of the background sink strength for this particular region, that the sink strength was degraded by about 50%. So the final thing then is in some sense a so what question. So I've just explained the water consequences. We've mapped those onto carbon consequences. And the idea then is to really understand what this means. We need to understand it in a temporal dimension. So what I'd like to do here is go back in the past and then go back in the future and to briefly explore how the likelihood, or better yet, how the return interval of such a drought has indeed changed. And our vehicle for going back in time is actually going to be tree-ring based reconstructions of summer PDSI. And it turns out that this particular product actually extends through the temporal extent of the turn of the century drought, such that it's more an apples to apples comparison. And the simple way to then benchmark the TCD is simply to take the mean value and then to look back in time and to see how many five-year periods meet or exceed that particular threshold. And when we do that, we have to go all the way back to the 12th century. That is, we have to go back prior to the year 1200 Common Era to actually find any type of event that meets or exceeds this particular threshold. The shorthand way of saying this is that the turn of the century drought was in effect an 800-year drought event. Moving forward in time, the picture actually changes. First off, we can no longer use tree-ring-based reconstructions forward in time. That, of course, doesn't really make any sense. But we can use precipitation data. And so what I'm showing you here is precipitation data from the CMIP-5. And what we've done here is we've stitched together the so-called historical experiment back to the year 1900 with a business-as-usual-like experiment out to the year 2100. And we benchmark the turn of the century drought in the exact same way. Here, of course, instead of using PDSI from tree rings, we're using precip. And what should be fairly apparent here is that the precipitation regime associated with the turn of the century drought, apart from the actual drought event itself, is completely absent from the historical experiment. However, we see as we move forward, what we are considering now an abnormally dry period is actually going to become very, very commonplace. And out towards the middle of this century, and certainly towards the end of it, what we are calling basically a very dry event is going to become the wet end of a new hydroclimatic state. So to briefly summarize, I've talked a lot about the 2000 to 2004 turn of the century drought in Western North America. What we've seen here is that this was actually the most extreme drought in the past 800 years, and that it very much elicited a large and spatially coherent drying out or drying down of the terrestrial biosphere, and it also degraded the land sink in the study area by about 50%. Just to put that number into perspective, that is the actual magnitude here, is somewhere on the order of 5 to 10% of U.S. fossil fuel emissions. And what we see moving forward in time is, again, what we were calling abnormally dry is actually going to become very commonplace, and towards the end of this century, it's going to become the wet end of what I would argue is a new hydroclimatic state. Thank you.
Time for one very quick question.
Yes? Correct? Yes. Well, what I've seen here is, one way to generate what you might call pseudo-confidence intervals in terms of using a multi-model ensemble is really to count the amount of models that show the exact same trend, right? And in this particular case, when you don't look at the MME, but you look at the individual models, 9 out of, I think, 12 show this very feature. So I think we can have a fairly strong confidence in this being where we're headed. Okay.
Thank you. Next up will be Trishan Yi discussing his perfect deficit approach.
Sorry, I would like to stand here to talk something. You know, I came from New York, and so the hurricane Sandy just visited New York City this year, and Hurricane Irene visited New York City in the last year. And a few tornadoes swarmed New York City in 2010. So you see, in the past 100 years, just less than 10 hurricanes have visited New York City. So the question is why extreme weather events become more frequent and more violent. So the theory tells us about this is because warming climate increases the water holding capacity of the atmosphere. So the water holding capacity is measured by saturation vapor pressure. The saturation vapor pressure is predicted by Clausius-Cronbron equation. Yeah. So atmosphere can hold more water vapor in hot weather. It's just like a large bucket can hold and dump more water. So hot weather, warming climate has a potential to produce heavy rain that increase the flooding. And also, you know, the more water vapor condensed into the liquid water, more latent heat is released. So the weather become more violent. And meanwhile, it takes longer to recharge atmospheric moisture. So the period between precipitation and precipitation become longer. That increases drought. So global warming that increases the change, the water holding capacity of the atmosphere. Yeah, and this needs more extreme weather. So the question is, this capacity change in the atmosphere would change another capacity. What capacity? The carbon storage capacity of the terrestrial biosphere. And this will feed back to the global warming by change atmospheric CO2 level. So our research is about how this extreme weather alter this carbon storage capacity of terrestrial biosphere. And we have here, we want to introduce a perfect deficit approach. This approach has been published, just recently published in the ERL special issues. Yeah. And the grasslands are most vulnerable to extreme climate events. The question is, how do we identify this drought effects on grassland potential productivity from daily precipitation data? Yeah. And we developed a new drought index, local dryness. That is defined by daily precipitation and daily net radiation in past 100 days with weight. So today, precipitation has maximum weight. Yeah. And we developed this perfect deficit approach with concepts of GPP and CPC and perfect CPC. And GPP, everybody knows that is gross primary production. So that is derived from the flux tower data. Yeah. And CPC is defined by maximum daily CPC. Yeah, just like Ichi talked about that, the maximum GPP in a day. And the curve under CPC represents carbon storage capacity. Yeah. And perfect CPC, for example, if we have 7 years data, so perfect CPC, that is defined by maximum CPC. You see 7 years, we pick up maximum one. So then we got a perfect curve. Then the difference between perfect CPC and CPC, we call it CPC deficit. Yeah, CPC deficit. So you see the triangle dots indicate the local dryness. You see this CPC deficit increases with increasing of local dryness. Has a very good dynamic relationship between this CPC deficit and local dryness. So yeah, this is the tower data, and this is for Canadian sites. Just to give you an example, we have analyzed several sites. This is just for 2003, this tower data. How about LEA data? We applied this perfect deficit approach to the LEA data. We can get perfect LEI and LEI. You see the deficit are consistent. The same story. How about 2001? That's the deficit from the tower data. How about LEI data? How about another year, 2002? So you see the deficit is in the spring. Yeah, the consistent story. And we can do something, integrate this CPC deficit that's equal to that area. What is the relationship between this CPC deficit and annual dryness, not local dryness? So this is growing season CPC deficit, and axis is annual dryness. It has very good correlation. This is for LEI. That's LEI deficit. That is annual dryness. Okay, conclusion. And the perfect deficit approach, that's just from different angle. We usually use average, then you see anomaly. Now we are different approach. Yeah. And you see the perfect deficit approach, the local drainage are useful for identifying the impact of climate events on grassland potential productivity. The carbon sequestration capacity of grassland ecosystem are severely reduced by extreme droughts. And this is just logical speculation. You see the grassland is declining, and that's maybe a result from the positive feedback between warming associated drought events and grassland CPC reduction. Yeah. If CPC reduction is, that means more CO2 go to the atmosphere, speed up global warming. Global warming causes more extreme weather. Yeah. So how about the first? We are doing about the apply this perfect deficit approach to forest. Here, just give you an example. That's a piece, a perfect CPC. That is the CPC. That is the CPC deficit. And another curve, 8 max. That's for the light response curve. That's you see. That's a CPC. Really indicate that it's carbon storage capacity. Yeah. And yeah, this is Amax. This is data. Yeah, we calculated about that. How about apply to this perfect deficit approach to global GPP? We can calculate the perfect GPP. Then we see deficit for each month or each day to the perfect. Then we got the map. We got a lot of map. Just to show you an example. Yeah. So we are working on this data analysis. Maybe report next year. And finally, this project is supported by ASF. Yeah. And another announcement. So, you know, we have extreme events and the carbon cycle special issues in the ERL. And it was deadline is December 31st. So I just asked the publisher to extend to another half year. Yeah, maybe another good news. You are welcome to submit your paper to these special issues related to extreme events and the carbon cycle. Thank you. Yeah.
Time for questions.
So you said that you're using 100 days.
Yeah.
Right? Precipitation and the radiation.
Yeah.
Like, to calculate the local dryness.
Yes.
Why do you use 100 days instead of 50 days or 30 days?
That's a very good question. Yeah. I think I have. No, yeah, I did not have one data. We have tests. We test the 100 days, 50 days, 30 days. They are very consistent, very consistent. However, for the 30 days, the amplitude is very large. And for the 50 days, the amplitude is large. For 100, amplitude is smaller. And when drought events come, they respond consistently. Not a big difference. Yeah, but less than 30 days, no. That's will be in this number will be going crazy. You know, maybe 10 days, maybe 30 days there's no rain. That will be go to the infinity if the time period is short. Yeah, we have tests that use the tower data. Yeah.
Well, so those dryness accumulate 100 days, but technology, 100 days were already cross seasons, right? For example, from spring to summer, right? Those vegetation respond already, right? Several months passed, and then how you, in other words, there's lots of long time lag already there, so we can between dryness condition versus the carbon matter already. If you do this type of correlation,
Yeah.
I think that
Oh, yeah. So we give the weights. Yeah. Today we give the linear weights. Today is maximum. The day before yesterday, for example, today give the weight is 1. The day after tomorrow 0.9. Maybe after the past 10 days, the weight is very small. So that's the one reason. Another, precipitation is input to the soil moisture, soil water. However, this plants respond to precipitation. That's by soil moisture. Soil moisture is the water budget. This water budget is determined by energy budget. There's a water budget coupling with the energy budget. So drought is a reaction of plants to the water. Precipitation only the input. Yeah. So I think local dryness is a very good indicator, just like soil moisture. However, soil moisture data, not many, right? Not available. So yeah. If you calculate the soil moisture, you must be using the model related to the vegetation, right? Yeah.
So one kind of issue is for estimating perfect CPC, you have to know the maximum daily GPP. So how many years of data do you need? Because you might in the future get something that exceeds that. Right?
Yes.
It must take longer than to get a good mean.
That's a very, very good question and a tough question. When in my paper published, the reviewer asked that question: how do you guarantee I use at least the 4 years data? That's I think a reviewer asked that question. Why you are guaranteeing this 4 years data? You catch up this, this precipitation data you catch up this drought events. So then I go into one paper, right? There's one published paper for more than 50 years precipitation data in Wyoming that I'm looking for. Precipitation never repeated. However, I did not find drought year more than 3 years. Yeah. So I think 4 years, that should be fine, at least. This perfect deficit approach is data-based nature. Yeah, we have data. However, we have data. If more years, the longer time, like Harvard Forest, that would be good. If we don't have, we can see something. Just like the drought, there's a normal, abnormal, right? Yeah.
All right, thanks, Tree. Our final speaker will be Paul Stoy, discussing photoperiod controls on photosynthetic capacity.
Okay. This is some work that I've done with Amy Trowbridge and Bill Bowerly. Amy's at Montana State with me. Bill Bowerly is at Colorado State. And because this is a FluxNet effort, I'd like to thank, of course, all of the individuals, organizations, funding agencies that went into FluxNet, as well as those who chose to contribute their data in the free, fair use manner. And I'd like to thank everybody here, of course, for staying till the hopefully not so bitter end of the entire conference. This work is following on a paper that Bill Bowerly wrote this year in PNAS, where he measured the VCmax, the maximum carboxylation capacity, and the Jmax, the maximum electron transport rate, of a number of leaves in about 23 different species, and found a very strong seasonal pattern in both that, which with a relatively nuanced variance decomposition analysis, was found to be more related to day length than temperature. Of course, there's some temperature dependency. This is an enzymatically regulated process One would hope that these enzymes, which are in fact function of temperature, would be related to temperature. But there's also reason to believe that a plant, when it knows when the sun is rising and setting, would have some chronobiological control over its own enzyme activities. So day length was found to be more important in describing the seasonal variability, which importantly was less related to common leaf spectral indices. These did not drop off so much in the autumn as opposed to the Farquhar parameters, the VC max and the J max, such that there might be an overestimate of these parameters in the autumn, leading potentially to overestimates of carbon uptake by the biosphere. And when you take the entire biosphere approach, running CLM, Peter Thornton and others perform this work, you find that when you add a photoperiod correction to the VC max term especially, you do in fact find significant differences between the model before and after that adjustment. Of course, predominantly in the higher latitudes where seasonality is more of a feature. After performing this correction, it has global implications. This is the detrended CO2 mixing ratio. I'm just choosing one site from Mauna Loa, but it worked globally. With the uncorrected CLM model, the one that introduced the VC max photoperiod correction had a better fit to the detrended mixing ratio at Mauna Loa and other stations. So my question is a little bit more simple. And that is, really, does temperature or photoperiod better explain the seasonality of parameters that are important for photosynthetic uptake, not necessarily VC max and J max, light response curve parameters, at the ecosystem scale? And the reason I wanted to pursue this is for a number of reasons, is that the parameters, or the inputs, the variables that we use to estimate something like a canopy VC max or a J max, all of these have uncertainty. So, we have things like air temperature, leaf nitrogen, leaf age, leaf area index. All of these inputs are uncertain. And I'll show you a little figure next describing why that's so important. But I also wanted to point out that these aren't necessarily new findings. For a long time, we've known that the VC max in this example has a relatively typical seasonal approach with some characteristic summertime declines, of course, after senescence. This is work by Wilson and others at Oak Ridge in 2001 on primarily oak trees, with also some deficits due to drought. And this reminded me, actually, after I put this together, Gabby Catul wrote an interesting paper in 2005 demonstrating that you can actually make VC max formally a function of water availability if you introduce a simple and logical model where the demand for carbon is balanced against the supply of water in the tree. So the Farquhar photosynthetic parameters actually become a function of the hydrology. So there's many explanatory variables that go into describing these ultimately enzymatically controlled processes. But there's also one thing, day length, zenith angle. This is the one thing, along with death and taxes, that we know really for certain in an ecosystem context. And the reason I wanted to point out the importance of zenith angle is that those inputs that have uncertainty introduce something that maybe we don't talk about enough as carbon cycle and ecosystem scientists, and this is the errors in X, the errors in driving variables problem. So this morning I was looking for a nice demonstration of this on Google. I didn't find one, so I had to do it quickly numerically over breakfast this morning. When you have a simple relationship between two things that are absolutely related, an X and a Y. You have some uncertainty in the Y variable, the explanatory variable. That, of course, will give you errors in estimating the slope, but they're not very biased, right? You have uncertainty that bounces about that slope, which, as you can see, is clearly 1. Now, when you introduce some uncertainty in the explanatory variable, the X term, the errors in X problem, you in fact will have a biased estimate of that slope. So introducing inputs that have a significant degree of uncertainty could potentially bias your understanding of the response, except when zenith angle is involved, because this is the one thing we know. So I took over 80 site years of data from over 400 site years of data from over 80 sites, excluding the dry and managed ecosystems whose seasonality are determined clearly by things other than day length and temperature. And I performed some simple analyses on these to get at this relatively simple question. So I asked first, is there information can we perform a Granger causality interpretation of the information that's contained in the day length time series or the air temperature time series as they respond, or as the gross ecosystem productivity or gross ecosystem productivity normalized by total light are responding to these drivers? And, as you might expect, almost all of the time, GEP is day length Granger causes GEP, but only at about a third of the sites does air temperature directly control that. After you normalize by the amount of light that falls in the first place, air temperature, in fact, does Granger cause normalized GPP almost all the time, as does day length, and these results are already arriving initially at my conclusion that both of these terms are important. So I also performed a light response parameter analysis. I fit a couple of different models to the PPFD, photosynthetic photon flux density, versus net ecosystem exchange relationship for all of these nearly 400 site years. Using here a demonstration, the Walker Branch for day of year 195, just a week in the summertime of 1999, and calculated our parameters. We have our beta, which is our Amax, which is our maximum NEE. You can add that to the respiration and get the maximum GEP, and then you also have the initial slope of the light response curve, our alpha. I'll focus mostly on the Mr-Lish model rather than the non-rectangular hyperbola that has an additional parameter here that describes the curvature. Can be relatively difficult to fit against noisy eddy covariance data time series. And I avoided the rectangular hyperbola because it tends to drift sometimes such that it thinks that when you have 1,000 ppfd, which you'll hopefully never have, your NEE is something like 100, which you'll never see. A leaf will be burning all the way up at that point. It's just a way to get a better handle on what that beta parameter actually is. And with that then, right, for different days of the year, with this moving window of parameter fits for all of these almost 400 site years, you can calculate the maximum beta value, or the different parameters. Also air temperature and day length, which we know to be consistently about day of year 172, excluding our leap years. And in this example, you can see that our maximum value of our beta parameter is falling shortly after the day length, but well before air temperature. This isn't actually always the case. When performing this type of analysis, it tended, right, our parameter values tended to peak actually after that of air temperature, suggesting at the ecosystem scale the findings of Bowerly are not maybe quite so simple. A number of other variables are at play. You can separate this analysis into biome or climate types. You can dig deeply and find some things, but to a first order, right, there's a relationship with latitude, as one might expect. A lot of these findings apply, of course, more to ecosystems that have more seasonality and light. And a larger finding here is that, right, what I did here is a simple information criterion analysis of very simple models for the variables that describe the different photosynthetic light response parameters, and found that in almost all cases the model wants both air temperature and day length as explanatory variables, independent variables to describe the parameters that we're interested in. So in conclusion, right, there's information in both day length and air temperature that help explain the seasonal variability of these parameters. Of course, we could look at more things, but like Trishong pointed out, getting soil moisture measurements across the entire FluxNet database has proven to be somewhat elusive. A number of sites have these observations, a number don't. And really what I was trying to get at with this analysis isn't so much that day length and or temperature and or something else controls the seasonal variability with these parameters, rather that there are certain things that we have that are, for most intents and purposes, almost entirely error-free. And if we can creatively use these inputs to help drive things in our model that we're interested in, we can ideally come up with models that have lower uncertainty, especially bias error. So I'd like to thank everybody here again, of course, all of the collaborators and contributors, and I may or may not have a couple minutes for questions. I intentionally finished a little bit early. Thank you. Trevor. Yeah. Yes, and I excluded it because it is extremely uncertain, especially at multiple sites, especially over many years. So yeah, leaf area index, right, a number of these things, of course, yeah, what could be more important than leaf area index? But there's also, you know, your radiation attenuation throughout the canopy, the fact that at the canopy scale you have light that is impinging upon leaves that have different inherent VC maxes and J maxes. And even in my example temperature, you know, I brought that up. You know, there's air temperature is so easy to measure, but what is the flux weighted average leaf temperature that is describing the physiological processes that Farquhar is describing in his model? That is not an easy question to answer at all. So even these variables that we think are relatively certain, or at least we're measuring them quite readily, it need not be the case that, you know, that it's actually an appropriate view of the physiological mechanisms at play. Radiation transmission to the canopy, all sorts of things going on. So I wanted to absolutely simplify the problem to its core, and then do some analyses on the outputs. Margaret has a question too.
Yeah, it seems like this would be a very nice proxy, but you know very well from many things like the area, to the extent the plant is well adapted to its site, because presumably summer happens at the same season in that site every year. But I wonder what you think about the asynchrony or the change in that with climate change. So it's a proxy for things for a well-adapted, locally adapted plant population.
Yeah.
I'm curious about your thoughts.
No.
Great analysis.
No, it's very true. Who's to say that, you know, a plant in a different environment, if you took a tropical ecosystem and suddenly moved it to the temperate zone, how it would deal with that seasonality? This is, you know, over evolutionary timescales, not something that ecosystem type has ever seen. So I don't have an easy answer to that question. It's really thoughtful. But you know, you'd think it would be important. I'm not saying that there's, you know, plants are like necessarily locked in or hardwired to respond a certain way, but you know, some of them are at a day length. There's a whole lot of enzymatic processes that we were almost never considering, but you learn about, you know, in your basic botany classes. And these chronobiological type mechanisms, I'm wondering if we couldn't be thinking about them, incorporating them somewhat better than we are, in that we tend to think about environmental drivers rather than, you know, plant enzymatic processes. If that makes sense. One more question. Mm-hmm.
If you look at something that has, so a conifer or a eucalypt or, you know, evergreen broadleaf will probably have a very different pattern.
Mm-hmm.
So that, like the conifers that I mentioned in France, in fact, VC max is highest in the middle of winter because the needles flush there towards the end of the summer, and VC max kind of ramps up as they extend, and then sort of comes down again during the spring. So it's more related to the age than it is to further period in some function.
Yeah, the interesting thing.
I don't know how that changes.
No, that's absolutely true. I mean, I put, you know, leaf age here as a factor, and they're different plant physiognomies. They have different seasonalities. But when I separated things out by ecosystem type, my answers were not very clear, keeping in mind that I'm not actually looking at VC max. I'm looking at something that's kind of related to J max. So so these types of analyses would be really cool. And I should also point out that Margaret Grunendijk and others have used the FluxNet database, and other studies have done this as well, to infer what the VC max would be. Now, these types of analyses require leaf area index and another number of other canopy scale variables to help get the best VC max estimate you can from flux data. So I couldn't agree more, but when I partition things, I'm only showing some, you know, climatological relationships here. When I did it by plant functional type, it didn't fall out quite as cleanly as I thought. But I guess that happens a lot with plant functional type as a explainer. Okay, thanks everyone again, and thanks for great AGU this year, huh?
Thanks, Paul, and thanks to all our speakers. That concludes our session, and that concludes the meeting. So safe travels, everyone, and good night.